The gene stays locked in the chromosome. The protein gets built somewhere else, so something has to carry the message between them. In 1961, 3 scientists caught that courier by weighing it. A courier that lasts only minutes, then vanishes. By 1960, everyone knew where proteins were made. Little machines called ribosomes, thousands to a cell. But how they worked was a guess. The leading idea came from Francis Crick himself. Each ribosome is a dedicated template. One ribosome, one protein. Its own RNA, the gene's stamped copy. Make a new protein, and first you build a new ribosome to make it. But there was a crack in that picture. Infect a bacterium with a virus, and within minutes it stops making its own proteins and starts making the viruses. Far too fast to build whole new factories. And years earlier, Vulcan and Astrachan had seen something odd. Right after infection, a burst of fresh RNA, short-lived, matching the virus's DNA, not the cells. A message, maybe? No one could prove it. So Jacob and Monod made the bold guess. The gene doesn't build a new factory. It sends a short-lived message to the factories already there, which posed a clean, testable question. After infection, does the virus build new ribosomes or borrow the old ones? The two answers look identical down a microscope. You'd have to tell a brand new machine from an old one reused. And that is exactly what Meselson could do. 3 years earlier, he had weighed DNA. He grew it heavy, let it copy, and read the answer off a density gradient. Now he turned the same balance on the ribosome. Grow the bacteria for generations on heavy atoms, nitrogen 15, carbon 13, until every ribosome in the cell is dense. Then infect and switch the food to ordinary light. From that moment, anything new is light. And tag the fresh RNA with radioactive phosphorus so you can find it. Now the trap springs itself. Spin the mix in a cesium salt gradient, and every particle sinks to its own weight. If the virus builds new ribosomes, the radioactive RNA rides down with them, light, high in the tube. But if it borrows the old ones, that same radioactivity clings to the heavy ribosomes and sinks. One tube, 2 possible heights. The whole answer is just, where does the red land? But the first tubes were a mess. The radioactive RNA wouldn't stay put. It smeared, it drifted, it refused to band with the ribosomes at all. For weeks, the experiment simply failed. Then, one afternoon, a colleague drove Brenner and Jakob out to the beach. And lying there in the sun, Brenner suddenly leapt up, shouting, "The magnesium!" The cesium was stripping the magnesium that holds a ribosome together. Starved of it, the machines fell apart and dropped their RNA. They raced back and flooded the gradient with magnesium. This time, the ribosomes held, and the counter told the story. The radioactive RNA, new, made only after infection, sat squarely on the heavy ribosomes. The old ones. No new factories had been built. The virus had sent its message to the machines already there. So the ribosome is not a template for one protein. It's a universal reading head. It will build whatever message is fed through it. And the message itself is a separate molecule, a working copy of the gene, made fresh, used, and destroyed within minutes. They named it messenger RNA, the missing middle of the central dogma, DNA to RNA to protein. What makes it beautiful is the economy. A whole theory of the gene overturned, not by a new machine, but by where a spot of radioactivity came to rest. The same weight trick that had weighed DNA, borrowed and turned on the factory. And a month of dead ends undone by a single ion, the magnesium. Sometimes the whole answer is hiding in a missing pinch of salt. Beautiful experiments.